Hello, and thank you for joining us on another episode of PlantGENE Podcast today. I'm Samatha Gunapati, a research lab supervisor at Washington University in St. Louis, WashU. So today I'm co-hosting with Aimee, project coordinator of PlantGENE. Aimee, can you please introduce yourself?
Sure. Thank you so much, Samantha, for that introduction. So like you said, I'm Aimee Malzahn. I'm the project coordinator of PlantGENE. And if you're new to the podcast or to PlantGENE, the PlantGENE project centers around the enhancement of plant genetic engineering to ensure a sustainable supply of food, feed, and fiber. In each podcast episode, we welcome esteemed leaders from academia and industry to share their experiences and insights for the benefit of our listeners.
Okay, great, Aimee. So today's our guest is Dr. Keunsub Lee. Keunsub is an adjunct associate professor in the Department of Agronomy at Iowa State University. His research focuses on developing and improving the plant biotechnology tools for crop bioengineering. So Keunsub constantly seeks innovative genome engineering tools for Agrobacterium strains and also has generated useful strains for plant transformations. He also shared the axotropic and recombination suppressed Agrobacterium strains with the research community. Keunsub is also a member of PlantGENE Steering Committee, where he creates and implements the vision for PlantGENE. So today I'm delighted to have this opportunity to talk Keunsub. Welcome, Keunsub. Thank you for joining us today for our PlantGENE podcast.
Thank you, Samantha, for your invitation, and thank you for having me. It's great to meet you virtually, and I'm glad to share some of my experience.
Great.
Thanks. So let's start with your research. Can you briefly describe your current ongoing research projects?
Yeah, sure. My research, two main goals is one is understanding agrobacterium biology and improving strains more suitable for plant transformation. That is one side. Another one is improving plant transformation method. For our major plants are maize and sorghum right now. We're also working on the soybean transformation a little bit. And my major goal is to improve existing methodologies, and including engineering agrostrain and utilize it for different transformation method, and also develop or optimize plasmid construct to make them more efficient for transformation.
For our listeners who maybe aren't as familiar with Agrobacterium, could you explain why Agrobacterium is such a useful tool for bioengineering?
Yes, Agrobacterium is actually closely related to the nitrogen fixing rhizobium species. So they are a symbiont of many legume species. And then for biotechnology, they have been utilized as a tool because it's considered as a natural genetic engineer, because it delivers, it's the only genus of bacteria which delivered its own DNA segment called the transport DNA or tDNA. And after discovery that whatever DNA sequence you put between the two border sequences, left border, right border, they will be delivered to the plant cells. So the community has been utilizing more than 40 years. So it's low maintenance. It does not require special instrument, right? So it's a bacteria, just like E. coli. We use it for cloning. You put your plasmid DNA that contains your gene of interest. It'll deliver. It'll do the work for you, hard work, to deliver gene into the plant cells.
Great. So you have a great experience engineering the agrobacterium. So your research focuses on mostly engineering the agrobacterium strains and implementing them for a transformation, right? So,
Yes.
but most of the crops are genotype dependent. So which that will prevent mostly like researchers from using the same protocol to transform several genotypes. So how this problem being addressed? How we can solve this problem?
That is a really long-lasting question. How do we overcome the genotype dependence? For many plant species that are recalcitrant to transformation, there are multiple layers of bottlenecks. If the gene delivery, such as tDNA delivery into the cell, is the bottleneck, we developed the ternary vector system. By combining compatible ternary helper plasmid, which carries extra copies of a virulence gene, it greatly enhances the tDNA delivery efficiency. So in that case, you can expand the transformable genotypes. For instance, we are using a quick-on method for B104 immature embryo transformation. With our ternary helper plasmid, the transformation frequency is very, very low because the DNA delivery will be limited. You need a longer co-cultivation time. But with the ternary helper plasmid, we just do overnight co-cultivation, and now our transformation frequency is very, very high, regardless of the immature embryo quality. Of course, immature embryo quality is the most important part, but even lower quality immature embryo, you can still achieve a transformation.
So you told like ternary, right?
Yeah, ternary vector systems.
Ternary vector system. Is this a vector system? Can we use like all type of genotypes, and it's only for and also how long it will take for the one genotype to another genotype? But because there are a lot of different maize, soybean example, for example. So you told like it takes time, and also how much time it will take for the subculturing and all.
Maybe I'm not sure if I took your question. So you are saying that what is the entire process?
Yeah.
With the quick-on method, it takes about 7 to 8 weeks from infection to rooted plant regeneration.
Okay.
That's pretty quick. And if the bottleneck is the DNA delivery itself, then you can utilize a ternary vector system. So to use a ternary vector system, you need to understand the plasmid compatibility, because now you're adding additional plasmid into agrobacterium cells. So to do that, you need to have a compatible origin of replication for agro, and then different antibiotic resistance gene, so that you can select both plasmid in agrobacterium using different antibiotic resistance. For testing different genotypes, it'll be best if you already have a regeneration protocol set up or a tissue culture protocol set up. Then you can utilize the ternary vector system, because we assume the bottleneck is the DNA delivery.
Okay, perfect. So I have just one follow-up question. So how much transformation efficiency did you get after using this ternary system?
That's a good question. Our older protocol without ternary vector, we were achieving 4 to 7% frequency, meaning that 4 to 7 transgenic T0 event out of 100 infected embryos. Right now, we are easily getting 30 to 50%. And when immature embryos are good, we are reaching 100%, near 100%. So it's really nice.
50% is a big number. Yeah, that's great. Yeah, I think my next question is maybe it's same only. I was about to ask, can you describe that ternary vector system and how the system will help to improve the transformation? I think you already told about this. And
yes. There are different ternary vector systems there. And then you can combine a third plasmid. So the first one is a disarmed TI plasmid already existing in the agrobacterium. And the second one, is a tDNA vector, which usually we use called as a binary vector. And the third one is you add a compatible helper plasmid, which carries the either VRG, or a new version contains all the essential virulence genes. For instance, our recent helper plasmid has all the essential virulence genes from agro. That means you can use it for non-agrobacterium strains, which does not have the disarmed TI plasmid. It by itself is complete set of virulence genes, so including the type 4 secretion system, and then the VRA, VRG two-component system to activate the virulence genes. So you can utilize them for non-agro strain for transmission.
Perfect. So my next question is, what are the thymidine axotropic strains and what is their role in the plant transformations? This is new for me. Yeah, I saw your one of the research paper like you published it. So there is a
Yeah. It was originally published by Corteva AgriScience. In 2010. They first published that generating thymidine oxotropic LBA44 strain, they can avoid the overgrowth issues. So what oxotropic means, there are different types of oxotropes, and then by deleting essential genes within the bacteria or the yeast, you can make them oxotropic, meaning that without providing the essential nutrient, they cannot survive. Instead of using antibiotics or fungicide, you can just don't provide a specific nutrient, and then they will not grow. So this way, oxotropic, especially thymidine oxotropic Agrobacterium strains, they require thymidine supplement. So by omitting thymidine into the media, you can easily get rid of Agrobacterium after co-cultivation period. So some plant species when that requires very long tissue culture and a long co-cultivation period, they can cause a lot of overgrowth issues. So therefore, using oxotropic strains, you can reduce the amount of antibiotics, and then there's a less chance for them to survive in the tissue culture, and also reducing the risk, releasing the Agrobacterium with your genome editing or recombinant DNA into the environment, because they are not likely to survive outside of the laboratory conditions.
So these will also help to some escapes. This can also maybe sometimes.
It's a possible. You hear about Agrobacterium contamination from the transgenic plants, because they can still survive on the surface of the plant, and then when you move them to the soil, because Agrobacterium is soil-borne bacterium, they can easily survive and propagate in the soil, and then can be released to the environment. So there is always a risk of that.
So what are the current challenges you are facing in your research? Like, for example, it can be a tool, it can be a technique, or it can be some unanswered questions?
All of them. We always encounter problems. The experiment doesn't go as you planned. And when you implement like a new CRISPR reagent, for instance, sometimes they show toxicity in Agrobacterium, right? In that case, you have to solve that issue. Sometimes the cloning procedure is not straightforward, and especially our new construct that have the baby boom bushel for leaf transformation, those constructs are really huge, and then sometimes it's not very straightforward.
Thank you.
Always things that can be improved upon, but that's the fun part of science, right? Is there's always a puzzle to work on.
Yeah, exactly. Always puzzle that sometimes it's a kind of anticipated. Some of them are, you suddenly just pop up, and then you have to solve it.
Yeah.
So I wanted to switch from talking about research into talking about your work with PlantGENE. So we mentioned this earlier, but you're a PlantGENE steering committee member, and you are one of the co-creators for the new webinars. We've called this series the Interactive Series for Plant Transformation Protocols. For our listeners who haven't had an opportunity to go either attend a live event or watch the recordings, definitely would recommend the recordings. But this series covers protocols in detail and gives a lot of time for audience questions. What kind of impact is this series having? And have you seen a pattern in the types of questions asked by the audience?
Yes, I think it's really useful, especially for beginners who are learning about the new method, because you can actually ask the details which cannot be found from the published protocol or original research papers. So those papers are missing the details, but we know that the devil's in the details. Sometimes it's small little things, know-hows. It make a huge challenges, or impact. And then it's not easy to get answers if you work alone, or if there's no senior members who know everything. But with this new series, I think that what I like the most is it's a community, it's a collaborative effort. People are really sharing their experience and their know-hows, and I think it does have a positive impact.
Thanks. And then I guess kind of a follow-up question, thinking about the details that are missing from protocols, kind of what would you like to see in written protocols? More details, like more photos? What would help improve the current
Well, it's
status?
how much detail you put into the protocol, still there will be some missing information there.
Mm.
So what I see is if you buy a new kit, it comes with a handbook, right? The handbook has all the information there, but then people will ask for a simplified protocol because it's not really easy to follow, right? So therefore, we need both. We need a simplified protocol so that you can easily follow the step. But when it's not working well, then you need the troubleshooting guidelines.
Mm.
So ideally, we can have some more detailed guideline books. But again, in that case, it's not easy to find your answer for your question. So I think that's why it's an interactive series. You can immediately get answers. Even if it's not immediate answers, you can at least come up with some kind of discussions that can be useful for troubleshooting.
Great, thanks. That makes a lot of sense what you're saying, that people are going to want a simplified version and a longer version. And I think it's really hard for researchers to be providing both of those for everything that they do. So yeah, I agree. I think the series has gone really well and we've gotten good feedback on it. So I see it's having a positive impact. And last question I had for you was, do you have any advice for the next generation of plant scientists?
I think I am the next generation. Well,
Or you know what?
yeah.
Maybe that's a better question. What's some good advice you've received?
Yeah, I'm one of the younger ones. Well, it's joking aside. I think it's important to pay attention to the details, right? Stay motivated, and then be interested in a lot of different things, so that you can get inspiration. So sometimes you can get inspiration from totally unrelated topic, right? So that can bring you to new ideas. And then second thing is sometimes it's good to catch the big picture instead of just jumping into the method of project, and try to get the big picture first, and then get understanding of that. And after that, you can pay attention to the details. Some people learn different ways. Some people learn from details first, and after a while, they can understand the big picture, right? But I want to see the big picture first, and from there, what is the niche I can do my best. But everyone has different learning style, so I try to learn that too.
That's
Great.
great. Thank you.
Great. Thank you for the advice for the new generation. Thank you so much, Keunsub, for joining us today. It was my pleasure talking to you, and I am hoping to see spread of your vision and improving the new tools and techniques and helping the future science society and the world. And it was pleasure talking with you. Thank you.
Yeah, thank you so much. See you soon.
See you.
Bye.
Bye. Bye-bye.
PlantGENE would like to thank the National Science Foundation for their support. To learn more, please visit their website at nsf.gov.